Hi, my name is Regan Hayward and I'm a postdoc in the Integrative Informatics for Infection Biology group at HURI. The title of my talk is Improved Quantification of Host-Pathogen Dual RNA-seq Experiments. I'd like to start off by discussing what dual RNA-seq is. So it's based on RNA sequencing where we have a host cell that's been infected by a bacterial pathogen. And both the host and bacterial reads can be captured simultaneously. Through bioinformatic methods, the sequence reads are aligned back to the originating organism. There are some challenges, however, which include reads which multi-map to both organisms and how they should be assigned. And to date, there has been little to no investigation of optimal methods for dual RNA-seq read assignment. To address this, we developed an automated next flow-based pipeline called dual RNA-seq, which implements multiple approaches to assign dual RNA-seq reads. Briefly run through how the pipeline works. So you have inputs of Illumina-based sequencing reads and the host and pathogen genome and annotation. After some quality control steps and pre-processing, there are 3 mapping and quantification methods you can use. After the pipeline's finished running, you'll have separate host and pathogen features and various RNA class statistics and reports. Of the 3 methods is the STAR and HT-seq, which is a more traditional genome-based approach, and we're introducing as part of the pipeline a transcriptome-based approach to assign reads using Salmon, which is a lightweight alignment approach. The main advantage of using Salmon is it has built-in functionality to assign many of these challenging reads using a model-based quantification strategy. We wanted to benchmark all 3 methods. So we simulated dual RNA-seq data from a publicly available dataset, and this was HeLa cells infected with Salmonella typhimurium. We initially ran the data through the pipeline to generate what we called our ground truth, and then we simulated common Illumina-based read lengths and library types. You can see from 50 base pairs to 250 base pairs, single end and paired end. We then ran each of these individual datasets through the pipeline, and we compared the results back to the ground truth to see how each method performed. For the host, you can see here, we can see that both Salmon methods outperformed at higher accuracy than STAR and HT-seq, and Salmon selective alignment marginally was better than STAR and Salmon combined. For the pathogen reads, we see more definition, particularly around Salmon selective alignment, which had more accuracy. And overall, we can see across the different read lengths and library types that Salmon was able to assign more host and pathogen reads with greater accuracy. So to ensure the previous results were not restricted to model bacteria, such as Salmonella, we examined other publicly available dual RNA-seq data containing more diverse bacteria. As you can see here, we've got Chlamydia trachomatis, Mycobacterium leprae, Streptococcus pneumoniae, and Orientia tutsugami. We've got the same trend with the 2 Salmon methods outperforming STAR HT-seq, and this is extremely evident with the Orientia dataset. So this is the public data, not the simulated data. We wanted to focus in on the underlying genes being quantified. So the plots show the quantification comparisons between Salmon on the x-axis and STAR and HT-seq on the y-axis. The dots are individual genes and the biotype is the colour. The Streptococcus dataset in particular had a large number of multi-mapping reads, which we can see quite evident here for this large proportion of reads closer to the x-axis in Salmon. For the Orientia dataset, Salmon was able to assign more reads to non-coding RNA, which are these green dots. And these green dots of the non-coding RNA were known to have lots of repetitive elements, which could be quite challenging to assign reads with a more traditional genome-based approach. So we're looking at the host reads now, and this graph is comparing the HT-seq versus Salmon and the number of assigned host reads across the 4 datasets I spoke about earlier. What's quite evident is the difference in the number of assigned host reads compared to comparing HT-seq and Salmon. Particularly for Streptococcus and Mycobacterium, both these datasets had lots of multi-mapping reads as well. hence why there's a large difference. Looking at the biotypes, we can see for the Orientia dataset, we see lots of non-coding RNAs, and Streptococcus and Mycobacterium, lots of protein coding, and again, these were lots of the multiple mapping genes. Here we show the enriched pathways from the underlying genes, exhibiting greater quantification using Salmon. And so the Streptococcus dataset, the enrichment is dominated by general cellular processes initially, but also includes infection-specific enrichment, which is further down the list and not shown here. On the right is Mycobacterium. We see more immune-based enrichment, including growth factors, immune signaling, and neutrophil-specific responses. So in summary, a lightweight alignment and model-based quantification improves the accuracy of dual RNA-seq quantification for both host and bacterial transcripts. And our pipeline can be applied to a range of host bacteria systems, as I've shown. The pipeline can be run locally or on a cluster, and it's part of the NF-Core project utilizing Nextflow. It's containerized for reproducibility, and it contains comprehensive documentation for initial users, but also for more advanced users as you become more familiar with the pipeline. I'd like to thank everyone in my group at Harry, and also a lot of the team members in NF-Core for their support. Thank you.